So today's talk is about how good is my solid form, and I'm going to describe and show to you how structural informatics can help guide solid form selection. So my talk today is going to talk about the CCDC and the 1.1 million crystal structures in the CSD, the tools that we have developed together with our industrial partners to address some of the challenges during the drug development process, and three case studies where we've actually applied the tools to address industrial challenges. And then what does the future look like? So, just a little bit about us. So, the CCDC, or the Cambridge Crystallographic Data Centre, originated in the Department of Chemistry from the University of Cambridge. It is now a fully independent institution, not for profit, and a registered charity. And our main objective is advancing structural science through data, knowledge, software. So, Olga Kennard was the founder and the first director of the CCDC. And together with J.D. Bernal, she believed in the value of collating scientific data in a central archive. And this is how the Cambridge Structural Database, or the CSD, began. So, it's actually Olga's belief that the collective use of this data would lead to the discovery and new knowledge which transcends the results of individual experiments. So all the structures in the CSD, if we collect them all together, allow us to gain these new insights and knowledge from the data. So that was back in 1965. So now in 2021, we have reached over 1.1 million crystal structures, and the database continues to grow in size. We continuously get people submitting to the database. And the CSD contains actually every small molecule crystal structure published in the literature. And this literature can come from all over the place. It comes from crystal structures, from patents, theses, structures published in university repositories, and each of these structures are enriched and annotated by experts. Really, honestly, the CCDC and the CSD together are truly a global effort. And it's when everyone comes together that we can get a database with 1.1 million structures in to help us learn and gain new insights. All right, so what's in this million plus crystal structures? So, I work at the CCDC, I use the CSD regularly, and for me, it is truly a treasure chest of information. It contains organic structures, polymeric structures, multi component systems, and the organic structures we have are further broken down into drugs, agrochemicals, and pigments. So, in addition to the crystal structure data, we have data on polymorph families, we've got melting points, crystal shape and morphology. All this is additional useful data that we have that we can combine with our crystal structure information. In addition to the structures I talked about before, there's a wealth of information about drug structures in the CSD, and you can easily access approved drugs using our drug subset. Now, if we look at the top 200 drugs by retail sales in 2018, you can see all the ones that are found in the CSD, and these are color coded green. Also, from a recent publication in Nature Reviews, small molecules led FDA, that's the Food and Drug Administration, drug approvals in 2020. So, a key industry which benefits from the CSD is the pharmaceutical industry. And this talk is primarily going to be focusing on that today. So, linking this to pharmaceuticals and drug product design and development. So, the two things that I'm going to be focusing on is the molecule and the solid form. And you'll be hearing more about that in my talk a little bit later. So, I've talked a lot about crystal structures and why they are important. But why are they important in solid form selection? Well, vast majorities of drug molecules are delivered in a crystalline state. So it's important to be able to purify the APA. Solubility plays a huge role. I mean, is Is the API soluble in the solvent of choice, ensuring chemical and physical stability, being able to control the particle size and mechanical properties? So selecting a solid form with optimal material properties is a key step, in a successful drug product development process. And all of this information we can start learning and picking up from our crystal structure. So again, so we've got data, and what insights can we learn from that? So hydrogen bond pairings are very important. They are directional, they're strong interactions, they're responsible for packing. And we know this from the literature from Etta, Desaraju, and Joel Bernstein. So donor and acceptor pairings are extremely important. So this is an example of a Pfizer molecule. Here, the OH is donating to the nitrogen. Now, if this donor acceptor pairing was different, we would obtain a completely different landscape. So non-hydrogen bonding interactions and intermolecular interactions are also important. So this is where we start to build up a picture about, are the hydrogen bond donor and acceptors pairings favored? Is my structure stable? And what does stability mean? Do I have voids? What can fit in those voids, and how and what interactions are actually taking place? And we can start piecing this information together with our 1.1 million crystal structures in the CSD. So why solid form matters, bringing it all together even more. Well, we have a famous case of ritonavir. There was an appearance of a previously unseen polymorph after the product was launched and in use by patients. This was extremely costly to put right. And not only with production and distribution costs, the human cost involved was quite significant. So this led to a sharpened desire within the pharma industry to have methods to predict the likelihood of polymorphism during drug development processes. So the question we have now is, using over 1 million crystal structures, is it possible to assess the risk of finding a more stable polymorph? So from data to insights and to health checks. So the context against which we can health check a drug candidate is by exploring the intramolecular geometry, the geometry of interactions, and the likelihood of these interactions, and comparing the API relative to the CSD. So we have, this 1.1 million crystal structures, millions of geometric parameters, all these interactions taking place, over 10,000 polymorph families. This helps us build a picture of how good or how unusual our solid form is compared to the CSD. So CSD-Materials, this is a toolkit providing structural chemistry insights to aid solid form risk assessment. It was developed with the aid of the Crystal Form Consortium, and these are 12 of the top pharma companies. So this initiative was designed to address industrial challenges in drug development. So when I say this toolkit has been guided or inspired by the Crystal Form Consortium members, that's because we've worked on real life challenges and issues to help address those by providing solutions using the tools that we have developed here. So the health check workflow. This describes how we can combine our informatics results with experimental data. So this leads to this confidence and reassurance and validation of our workflows. Are we doing the right experiments? Could we do something differently? Do I need to include an extra solvent screen here? Is my torsion unusual? So we can start answering those kind of questions. So the health check workflow actually looks at the crystal structure. So your crystal structure helps us answer questions on space group, our chirality, Z prime. We're looking at voids and disorder. So we can learn all that information pretty much really early on. So is my space group usual? Is it unusual? How does it fall relative to the CSD? The next thing we would then look at is our geometry check. the bond lengths, the bond angles, the torsion angles. How do these all fit? Do they look usual or are these unusual? We then start looking at intermolecular interactions, our donor and acceptor pairings, the likelihood of functional groups and coordination. Does, for example, an NH2 want to coordinate once or twice? What's usual and what's unusual? And then statistics on all of these aspects. They help us actually recognize unusual and identify this opportunity, which helps aid the decision-making process in drug development. We are actually reinsured or empowered to plan the right experiments, or it's business as usual and your workflow follows the normal procedure. So why apply informatics? You know, why would you do this? Why do you think I'm already in the lab, I'm doing these experiments? What can informatics show you? Well, firstly, the tools are extremely straightforward to use and they return back answers very, very quickly. If you are collecting experimental data anyway and you have that information, we can use that to, you know, help develop a deeper understanding of your API, identify any weaknesses that you may have, reveals opportunities for intervention. It feeds into this whole risk management of solid form selection workflows. Now, it doesn't replace the experimental work, but it works alongside and in parallel with that. And this is an approach which has started to be used as standard by a number of pharmaceutical companies. So a lot of the work and data we use is actually proprietary. So it's really nice when we have an example that we could work with. So this is a paper with Pfizer that we have written addressing some of the challenges experienced during drug development. So today I'm going to talk about 3 case studies briefly, which use the structural informatics tools in the CSD-Materials toolbox and the health check methodology to address some of the questions that arose during this experimental work. So the first case we're going to be talking about is Maraviroc. So Form A showed 2 triazyl nitrogens accepting from an amide donor. Now, is this unusual? We noticed that the hydrogen bonding seemed a bit long. And again, off we went. We searched again similar interactions in the CSD using Conquest. And Conquest enables search and retrieval of information from the CSD. So actually, when we used Conquest, we realized that the geometry of this hydrogen bond was flagged as unusual. It was actually bifurcated. You can see here where Form A was. It's actually bifurcated and it was quite long. It says 3.2 here and 3.22, and this is our bifurcation showing a little triangle here. So immediately, as a result of this, further experimental work was triggered, and a second form then, Form B, was found. And now in Form B, you can see that only one of the triazyl nitrogens acts as an acceptor. And actually, the hydrogen bond now, if you notice, is around the 3 mark, was actually shorter than in Form A. And this actually matched CSD statistics much better. You can see from the mean from this chart at the bottom here that it falls nicely within CSD statistics. Form B actually turned out to be the thermodynamically stable form. So here in this case, we were able to trigger additional work, additional experimental work, resulting in a more stable Form B. And this was a direct result of the analysis that we had done using Conquest. The second case study I've got here is conformational polymorphism. So the Pfizer pain candidate crystallized in 2 conformational polymorphs, metastable A and stable B. Now, in Form A, you can see that the amide carbonyl is trans to the pyridyl nitrogen, whereas in contrast, in Form B, the amide group is more out of plane, and the amide carbonyl is cis to the pyridyl nitrogen. So here, the dominant geometry is found, you can see in this histogram here, around the 180 mark. And actually form B shows an unusual orientation of this group around the 35 degrees. Now, you can't see this on the scale, I apologize, because it's barely visible, but there are actually 6 structures which adopt torsion values of less than 45 degrees. Now, also, because of this orientation of these nitrogens and the oxygen, the hydrogen bonding is different in both forms. So in the 6 structures, the hydrogen bonding is the same as in form B. So the 6 structures here that you can barely see actually match the same hydrogen bonding that is observed in form B. So we need to consider these conformations in the context of how well they satisfy the intermolecular interactions as well. So what does this hydrogen bonding look like? So carrying on with our case study here, you can see we then went to explore how well these intermolecular hydrogen bonds are satisfied. And we conveniently did this using full interaction maps. And full interaction maps, we can actually compare the expected interaction geometry for hydrogen bond donors and acceptors to the actual packing shell of the 2 polymorphs. Now, if you look at Form A, the packing shell actually does not satisfy the expected interactions as well with regard to the NH2 amine position. There you go. Yeah, so really this little blue blob here should be sat on the nitrogen. So this would suggest that it is unusual and a suboptimal arrangement. Now, in contrast, if you look at Form B, the packing shell is beautifully satisfied with the expected geometry for both the hydrogen bond donors and acceptors. Now, this indicates a more likely and hence a stable arrangement. So although the torsion was unusual, the hydrogen bonding seems better. So in this case, the better hydrogen bonding actually compensates for the unusual conformation in 4B. It helps us explain and understand that to have this nice packing arrangement with the hydrogen bonding, it has to be twisted in that conformation. So actually, it's okay. So in our final example, which is crizotinib, so this was a Pfizer drug used to treat non-small cell cancer. Here we use the whole health check methodology. So we explore intramolecular geometry using MOGL, and we found that the database distributions, which were the bond lengths, the bond angles, the torsions, were all similar for functional groups. The packing environment, the intermolecular hydrogen bonding, as we explained using the full interaction maps, showed a very good match to where the expected hydrogen bond donors and acceptors were supposed to be sat. So here we actually now introduced hydrogen bond propensity. Now, hydrogen bond propensity generates potential hydrogen bonding networks and ranks them in terms of propensity and coordination. So now these are calculations were carried out to calculate and rank the likely donor and acceptor pairing combinations. Now, if you see in this little chart at the bottom, you can see donor N1 donates to acceptor N3, and it has a propensity of 0.72. And the donor N1 donates to acceptor nitrogen again, but it's a different acceptor, the NH2N2, and it has a propensity of 0.53. And these are the top 2 interactions that are observed in crizotinib, and you can see that they're actually observed in the crystal structure as well. So this is the likely interaction you will see, and it's also observed as well. And this chart here combines the mean hydrogen bonding propensity and coordination. So this little square, you can just make out in the corner, bottom right hand, it's actually pink. This is crizotinib. So this is our crystal structure of crizotinib. And you can see that there are no other triangles that are no other squares that are close by to that pink spot. And this suggests that there are no other hydrogen bond networks or coordination scores that are actually better than the observed coordination and propensity of crizotinib. So here we can confirm that an alternative stable polymorph is unlikely. So this actually complements our experimental work that was carried out, because for crizotinib, lots and lots of screening actually took place. Extensive amounts of screening took place, and it proved to be a monomorphic system. Again, combining this with the informatics, we can be confident that that's true, because we're not seeing anything else better in both propensity of coordination. So it has low risk of polymorphism. So bringing all of this together, we can see that the case studies actually highlight the evolution of our approach to structural informatics in understanding the solid form. In the first study, you see how structural analysis was able to identify a metastable polymorph of Maraviroc, and this mitigated solid form risk at a very early stage in development. In the second example, we use the approach to understand conformational polymorphism and how a better hydrogen bonding network can overcome the unusual torsion. And in the third case study, we demonstrate how all the health check methodology and combining the informatics tool complement the experimental approaches and give us that confidence and reassurance in the solid form of crizotinib. And this was actually in clinical phase trials were taking place then, so it helped us, really be sure what we were looking at was right. So these case studies allows us to use these state-of-the-art tools and look at structural informatics and the health check and how to actually, truly complement the experimental workflow and flag up potential areas of concern earlier on. So we've looked at solid form, and here what we've tried to show is how structural knowledge is used to inform key decisions in the pharmaceutical development processes, and how we can integrate these digital workflows to guide solid form development, identify risks early on, and actually we can have inclusion of proprietary chemistry. So if you do have your own in-house databases or your own crystal structures, we can run the tools against that for a more reliable prediction. So if there's any chemical space that is missing from the CSD, but you may have, we can capture that more reliably using the tools. So from solid form informatics, we've got the molecule, we've looked at the solid form, and the missing piece now is the particle and the properties which will help us lead to the product. We're looking at particle informatics. So here with this, this is ongoing research projects. We're developing new descriptors to better understand and model phenomena such as surface wettability. And approaches to problems such as these are typically used computational approaches. So combining these computational approaches with informatics methods can actually lead to novel and efficient solutions to these complex problems. And this was work that has been taking place with ADOPT, which is the Advanced Digital Design of Pharmaceutical Therapeutics, and is now being further developed because we were awarded an Innovate UK Knowledge Transfer Partnership grant to explore this area further with the University of Leeds. So if we bring it all together, this holistic approach in drug development, we've shown that by using crystal structures and the data, we can bring all the tools together. It actually, reveals this opportunity for intervention earlier on. It allows more informed experimental design to probe the risks and opportunities. And, we can start then linking these structures to properties and molecule and materials design. It's this minimizing of experimental footprint, the cost reduction, and strengthening your regulatory submissions. And if we're collecting this crystal data anyway, you're looking at your powder patterns or you're looking at your single crystal, it's making the most of the data that you have and what we can learn from it. And all of this, ultimately feeds into this scientific ecosystem to realize this vision of digital design. And this leads me to the end of my presentation, and thank you all for taking the time to listen. Thank you very much.